So, Optogenetics startup Prolific Machines raises 55 million Series B1, uses light to control virtually any cell function in any cell type. So, Prolific Machines, a California-based startup harnessing light-sensitive proteins to control cells, used in biomanufacturing, has raised 55 million in the Series B1 round led by Hitua Foundation, the corporate venture arm of New Zealand dairy co-op Fonterra Cooperative Group. The first close of the round, which takes the firm's cumulative funding to $86.5 million, was supported by Breakthrough Energy Ventures, Mayfield, SOSV, Shorewind Capital, Darko Capital, Conti Ventures, Continental Grain Company, and IQT, and several others. Founded by stem cell biologist Dr. Denise Kent, CEO, physicist and biomedical scientist Dr. Max Huisman, CTO, and computer scientist and ML engineer Declan Jones, CIO, in 2020, Prolific Machines introduces light-sensitive proteins to cells that are used to biomanufacture everything from vaccines to high-value ingredients used in infant formula. Rather than controlling cell function by adding expensive growth factors or other molecules to cell culture media, as companies do now, Prolific Machine exposes cells containing light-sensitive proteins to light at specific wavelengths, a technique known as optogenetics. This can activate or deactivate certain genes, proteins, or cellular pathways, delivering unprecedented cellular growth. CEO Dr. Denise Kent tells Ag Thunder News. This is the first time light's ability to control cellular function is being used outside of research labs to make everyday essentials. It's incredibly exciting and the start of something very big for lots of different industries. Dr. Maximilian Hörner, head of Optogenetics, Prolific Machines. Transitioning from using molecules to control cells to using light. For decades, says Kent, we've been using molecules, whether chemicals or proteins, to control cells, and they come with a bunch of problems. In some cases, the issue is cost, he notes, with some growth factors used in therapeutic proteins, IGF and EGF. Stem cell cultivation is FGF and TGF beta. Tissue engineering is VEGF and PDGF. Vaccine production is GM-CSF and IL-2. And gene therapy is FGF2 and HGF, coming with hefty price tags. But this is not the only problem, claims Kent. You can't control where these molecules go and when they go, where they go. We're just dropping them in bioreactors and hoping for the best. Another issue is sterility. Any component added to a bioreactor increases the risk of contamination. Plus, there are reproducibility problems because some of these molecules are biological in nature and so they are not consistent. You can buy FGF2, fibroblast growth factor 2, a widely used regulator in cell development, differentiation, regeneration, senescence, proliferation, and migration, from the same supplier with the same catalog number, but it won't necessarily be the same as the FGF2 that you bought a month ago or a year ago. Light is cheap, inherently sterile, reproducible, and can be controlled spatially and temporally, he adds. Every one of those problems is solved with light. The cheapest possible input into biology, plus it's easy to control both spatially and temporally. That is, you can control where it goes and when it goes, where it goes, which allows you to get higher yields. Controlling on the space axis also allows you to create patented products that other people won't be able to make using molecules that are moving around randomly. Light is also inherently sterile, eliminating contamination risk. Introducing molecules to bioreactors always carries the risk of contamination and is inherently reproducible, tackling the inconsistency problem, he adds. If you have 450 nanometer wavelength light today, it is the same as it will be tomorrow and 1,000 years from now. Another advantage of using light rather than molecules added to cell culture media to direct cellular activities is that it can potentially reduce downstream processing and purification costs post-harvest, as there are fewer media components you need to ensure do not end up in your final product, he says. On the regulatory side, meanwhile, there's a number of advantages of using light because it doesn't carry the same potential risks of chemicals or recombinant proteins, claims Kent. We've spoken to the FDA about this on several occasions, and they were very happy about the fact that we have a solution to eliminate the need for these other inputs. Breakthrough innovations like Prolific Machines' biomanufacturing platform don't come along often, particularly innovations that have the potential to unlock profound impact in both human and environmental health across multiple industries. Komal Mistry Mehatdaf, founder, director, Kitua Foundation in New Zealand. How it works. According to Kent, we take light-sensitive proteins, of which there are thousands, and we genetically attach them to different targets inside the cell. You can do that by making permanent genetic changes or via transient genetic changes, which would impact whether the cell would be considered a GMO for regulatory purposes. Targets inside the cell could be anything from a receptor on the surface of cells, an enzyme, a transcriptional activator, something that turns one type of cell into another, or something that tunes cells' metabolism to make more ATP, which provides energy for biochemical processes, and less lactic acid, which can limit cell densities per molecule of glucose. And you can toggle the activity of that target using light. What we've invented is a way for machines to control some cellular biology specifically for the first time. As the mammalian cells that Prolific Machines is focusing in are not naturally light sensitive, the only things that respond to light in the bioreactors are the targets that it has tagged with light sensitive proteins, enabling greater precision, he says. We have hardware that illuminates the cells. And then we have software that is taking all of the spectral data that we're generating and then learning using machine learning what's the right pattern of light to apply. We don't just leave the light on all the time. There are different patterns of light to do different things. For example, he says, with traditional approaches to biomanufacturing, we often ask cells to do multiple things at the same time, such as grow and produce protein. With light, it is easy to separate out those phases, which might be more efficient, he says. You can use one color of light to accelerate the growth with no protein production, and then switch to a different color to end growth and only focus on production. So you can reduce the amount of time it takes to get to maximum cell density and improve titers, the amount of an expressed target molecule relative to the volume of liquid. Initial focus on mammalian cells. To date, Prolific Machines has focused on mammalian cells, although there is no reason to believe the tech wouldn't work with microbial cells used in precision fermentation. For example, says Kent, who says the company will be announcing commercial partnerships in due course. This is in part because there's a lot more investor appetite to do mammalian applications than there is to do microbial applications, because you can make a lot more money. So we've initially focused on things like antibodies and cytokines, vaccines, viral vectors for gene therapy, and tissue engineering. Asked if anyone else is currently deploying optogenetics in biomanufacturing, Kent says, as far as we know, we're the first company to use light in this way to aid biomanufacturing, but there are other companies that using light in other contexts. For example, I'm aware of a company that is using light to reverse blindness by genetically engineering the cells of your eyes to become light sensitive. There's also a lot of activity in the neuroscience field. Prolific Machines has 14 patent families, 11 of which are owned by us, and then 3 of which are owned by universities, Stanford, Berkeley, and Johns Hopkins, he says, but we have exclusive licenses to them. Implementing the technology. Think of us as the NVIDIA for biology. Prolific Machines technology has been intentionally designed in a way that makes it very easy for people to use, says Kent. For example, the genetic constructs do not need to be in our cell lines. We've designed them in a way that can slot into anyone's cell lines, because some people have spent years optimizing their cell lines. Think of us as the NVIDIA for biotechnology, where we can elevate our partners' existing cell lines and product approaches, providing a critical infrastructure layer for biology. Similarly, specialist bioreactors are not required ads. All they need to do is buy our hardware that slots into their existing bioreactor infrastructure. The software is embedded in our hardware. To expose the cells to light, many bioreactors already have glass portholes through which light can be directed, he says. And as cells are being agitated, you don't need to illuminate 100% of the bioreactor to ensure that 100% of the cells are illuminated, he explains. We're also building internal illumination systems, so you could have lights on rods that go into the bioreactor, lights on the impeller, which stirs or agitates the contents of the bioreactor to ensure all cells access the nutrients, or you can add neutrally buoyant balls containing LEDs floating around in there. So, Prolific Machines' photomolecular biology platform uses light to guide cellular behavior and optimize performance over time using AI. According to the company, light can turn up or down important cellular processes, allowing precise control over exactly when and for how long bioprocesses like gene expression, receptor activation, and enzyme activation are active. So, cultivated meat, a far more elegant approach. Cultivated meat is one use case for Prolific Machines technology, given the potential to eliminate the use of pricey cell culture media components, says Kent. Well, some firms in this space are now using CRISPR gene editing techniques to edit cells such that they don't need to use certain growth factors. There are drawbacks to this approach, he claims. If you're using CRISPR to get your cells to express growth factors themselves, your cells are constantly pumping out a lot of growth factor, and you can't tune that out, whereas with lights, you can very precisely tune out how much growth factor activation you want at different times. The other problem with the CRISPR approach is that if your cells are constantly producing their own growth factors more than they need, you're putting a big metabolic burden on the cells, which slows them down. There is a sweet spot, and it shifts over time depending on what's going on in the bioreactor. So using light is just a far more elegant approach. End of article. So for this article, the most interesting part, in addition, of course, to the fact that they are already on a Series B1, they have raised $55 million in this round. So yeah, Breakthrough Energy Ventures. If we check the list here, our proprietary list. Yeah, Breakthrough Energy Ventures. Kirkland, Washington. In addition to which other companies? We have, yes, Mayfield, SOSV, Shorewind Capital, Darko, Conti Ventures, and InQtel. All part of this Series B1. So most of these are stateside, New York City, Philadelphia. Shorewind should be San Francisco. Darko is Philadelphia. Continental is New York City. Okay. But it's actually a hardware play. It seems from this article that yeah, it seems that the hardware is actually, we have hardware that illuminates the cells, okay, as illustrated in the photos in the article. And then we have software that is taking all the spectral data that we're generating. So actually, I think this is actually quite a good business model. So similarly, specialist bioreactors are not required yet. All they need to do is buy our hardware. So that's the initial sale for the business model that slots into their existing bioreactor infrastructure. All right, so an augmentation of the assembly line. The software is embedded in our hardware. All right, so the software and the lighting would be proprietary and protected by IP, but the units themselves are initial. So I really, really like this business model, given the fact that it's both software and hardware, and there's IP. So well done. And the last part of the article where it mentions about the CRISPRs, because that seems to be the incumbent, is that CRISPR in itself is now an antiquated and a bit dated, given the fact that CRISPR is sort of all or nothing. But then by using light and AI to determine what is best and what is optimal, then you can even increase and turn on and off certain growth aspects that are either needed or not needed at that point in time. So as he said, it's a more elegant approach and a more a la carte approach, if you will. So very well done. And it was very interesting from the fundraising side to see who was part of this exciting fundraising round for Optogenetics specifically. But this is agri-food tech. Okay.